who is a PhD student with Dr. Michael Conover in the Wildland Resources Department of Utah State. Maureen is originally from Colorado, and she started her Aggie schooling at the Texas Aggie School. But everybody know A&M?
Yes,
for
No,
really. What does it really stand for?
Agricultural
Agricultural and mechanical. They were the Aggies. Are they still the Aggies?
Yeah.
They're still Aggies. So once she got done with her bachelor's degree, she came up to the other Aggies, the original true Aggies at Utah State, and is currently getting her PhD. She's also a presidential doctoral research fellow, one of the elite researchers at Utah State. And with that, I'll turn it over to you.
Thank you, Frank. Thank you everyone for being here today. This was a great turnout. I guess maybe everybody just kind of got blown in here this morning. But so today I'm going to be covering part of my dissertation work on Great Salt Lake about the staging ecology of migratory water birds, and in particular focusing on birds that are using the open saline waters of Great Salt Lake. Before I get started, I would like to thank, first of all, my advisor, Mike Conover, as well as all of my committee members, especially Frank, who suggested that I come down here and give this talk today. All of the Great Salt Lake Ecosystem Program, they're all represented right over here, and none of this work would be at all possible without them. They're fantastic to work with. As well as our DWR pilots who have ferried us safely across the lake many times. And then many of you know Tony Roberts was the student on this project before me, and he really helped me get started with a lot of this work. And then various technicians and volunteers that have helped me out over the years. My work is funded by DWR, who I would also like to thank for hosting me today, and then also by USU's Office of Research and Graduate Studies, and then also thank the Ecology of Migratory Waterbirds who provides additional support. So I'm talking about birds that are using the open saline waters of Great Salt Lake. So we have tons of birds that come here every year, and a lot of those are using wetland areas, or we have various shorebirds, but this research is focusing on those species that are using the open waters out in the main part of the lake. And there's not as many species that use those areas because of the challenges of it being salty. And so in particular, I'm talking about Wilson's phalaropes, redneck phalaropes, and eared grebes. And each of those species represents a major portion of their continental population here. So we have over 1 million Wilson's phalaropes, over 500,000 redneck phalaropes, usually at the peak of the staging period, and then 1 to 4.5 million eared grebes. And for eared grebes in particular, half of their continental population is here. The other half is at Mono Lake. In past years, we've actually had more than half of the total population here at Great Salt Lake. And so even though they've got a really strong population size and their numbers are pretty steady, it's a management concern just because they're really relying on these two different areas for staging. Most of you probably know what Great Salt Lake looks like, but I wanted to use this image to show the 3 different habitats that I'm kind of focusing on and the differences there. So first off, Carrington Bay. Phalaropes spend a lot of time north of Hat Island, and this is right now really characterized by these exposed bioherms that as the lake levels have fallen, that's this light brown in the image is exposed bioherms, and then kind of some shallow water near them with submerged bioherms, but then it drops off. There's a shelf there that drops off into deeper waters. And so the birds are mostly congregated along that northern part. Grebes also use Carrington Bay before they migrate. And then Gilbert Bay, deeper, again, this image was taken kind of from over on the border between Gilbert and Ogden Bay, so there's some bioherms there. In the deeper parts of Gilbert Bay, it's simply underlain by, at times, deep brine layer or just soft sediments. And then finally, Farmington Bay, which is hypereutrophic and very shallow. Ogden Bay was a part of this study in the first year, and then because of low lake levels, we lost our ability to access that part of the lake, and also phalaropes stopped using that part of the lake. So grebes still use some of the parts of Ogden Bay, but I'll include Ogden Bay in some of the results and not in others. And it's just because we lost access to be able to get over there. But Farmington Bay has shrunk, but right now it's still there, and so that's it's shallow and characterized by having more algae than the rest. This research started because of the importance of brine shrimp, and then just trying to understand the role of these invertebrates in the ecosystem. So the reason we have all of those birds here in such large numbers is because they're here to fuel up before they leave for their migration south. And so, starting this out, previous research had shown that eared grebes are eating brine shrimp, so we had that established. And then we know there's all these brine flies, and we were pretty sure that phalaropes were eating those, but we didn't have a good idea of, for phalaropes, were they ever using brine shrimp? Are they just using brine flies? How are brine flies distributed across the lake? And kind of how those different resources play in, and whether there are other resources also that phalaropes are using while they're here. And then the lake is used for many human needs, including mineral extraction, shrimp cyst harvest, and then water, which has become more of an issue in these past couple of years as we've been in drought. And so water that is being used by the Salt Lake City metro area does not reach Great Salt Lake. So I wanted to share broadly my dissertation objectives to kind of give an overview of how this project will go, and then today I'm really going to focus on objectives 1 through 3 and 5. So we're looking at those invertebrates to start from there and say, how does it affect these other things? So when we're out collecting samples, I like to say we do all invertebrates all the time. Wherever we took samples, we took invertebrate samples, which means we have a whole lot of invertebrate samples, which also means, unfortunately, that not all of that data has been processed. So those, even though they're kind of the base of this, is not the data that I will be including in here today, but rather the rest of the portions of these objectives. And so the things that we're looking at here, in terms of phalaropes, again, previous research has been done on eared grebes, so this is a smaller portion, whereas phalaropes were a lot more unknown. And so we're really looking at their habitat use, their behavior, and their prey, and then their physiology, especially to compare between species and over the span of time that they're here. Eared grebes' physiology changes when they get here. They lose their ability to fly temporarily when they arrive at Great Salt Lake, and their digestive organs increase. Their flight muscles atrophy, their digestive organs increase so that they can take advantage of the prey. Whether anything similar to that happens in phalaropes was unknown, and that's part of the physiology part of this study, but also at Mono Lake there have been observed differences in Wilson's phalaropes versus redneck phalaropes as to what kind of prey they're eating. So we also really wanted to look at physiology to compare between those species to see whether that could give us any clues as to why that's different. And then finally, part of the eared grebe puzzle that we're still working on is just to try to figure out why they're leaving for migration when they do, and so that's that 5th objective. But so for these first 3 in terms of phalaropes, I wanted just to summarize it this way. We're really just basically looking at where have they been and what have they been doing, and also what are they eating while they're there. So first to talk about where they're spending most of their time. We conducted aerial surveys every other week throughout the summer, and we were not as interested in enumerating different phalarope flocks as to looking at where are the big flocks, and then where are there areas where there are not any phalaropes, and comparing the characteristics of those sites to see what differences we could see as to maybe why phalaropes are using some sites and not others. And we split the lake into west and east. This was based on accessibility by boat. So I would fly and figure out where the phalaropes are, and then this is part of where the GSLEP team was instrumental in that they would have boats waiting. We had a big boat for the west side in the deep areas, and the airboat would go to the east side, Ogden, for 2013, and then Farmington for all 3 years. And then whenever we went, we would do an invertebrate sample. So I just wanted to explain these a little bit. For a water column sample, you can see we used really fancy equipment, like a Lowe's bucket and a Subway cup, and we sampled all the water within half a meter of the surface. And this is because when phalaropes are foraging, they spin, and many of them will they can upwell prey from half a meter, and so that's kind of what we wanted to sample our prey from. And so in the shallow areas like this, we would take a stovepipe sample, and then we'd do vertical tows in deeper areas where it was too deep to use the stovepipe sample. Then we also needed to sample brine flies, and this posed a little bit more of a challenge because sampling flying invertebrates over water, especially in the deep areas, was a little challenging. So we tried a few different methods, but what worked best was putting sticky paper on kickboards, and this method brings us, I don't know, what do you guys want to say, joy? Laughter, because the kickboards are just a little bit different than what you typically see when you're sampling. So we set sticky paper out on the kickboards and floated them behind the boat because brine flies are artificially attracted to surfaces, so we wanted to get them out of the way and use those not really to get an absolute measure of brine flies, but to get a relative abundance of brine flies across the lake. And then finally, we were taking other environmental characteristics at each of these different sites to compare. So this map shows phalarope locations over the 3 years of the study, so 2013 through 2015. And the different sizes of the dots are representing different sizes of flocks. So I said earlier we were really looking for where those large flocks are. But at the beginning of the season, sometimes we would only find smaller flocks, so we mark those too. And at the end of the season, same thing. So this is just kind of an overview of where they are. So you can see, especially the Farmington Bay and Carrington Bay are really important areas for phalaropes. And so if I zoom in on these and then break them apart by year, Carrington Bay is pretty similar across the 3 years. So we can see again these areas north of Hat Island, and then on the west shore of the lake are really important areas. We see lots of big flocks there. Something to note really quick, I used a different lake outline for 2013 just to kind of indicate we had a little bit deeper lake that year than the 2 subsequent years. But overall, not too much difference between these 3 years. That's not what we see in Ogden and Farmington though. In Ogden Bay we have phalarope use of the bay quite heavily in 2013. We had some of our biggest flocks in Ogden Bay. And then in 2014 and 2015, as I mentioned earlier, they stopped using those areas for whatever reason. The resources that were there that they were using in 2013 became unavailable. So that's one important change. The other important change, and you can see this a little bit in the differences between the lake outline, 2013 we had more Farmington Bay. In 2014 and 2015 we had a lot less of it. And again, this outline for 2015, it actually shows a little bit more water than there actually was. And so you can see we go from flocks pretty spread apart to more grouped in 2014 and even more clustered in 2015. So they definitely were pulled together. They wanted to keep using those resources there, but there was just less habitat available as we had less water. So then moving on into what they're doing in these areas, what they're using there. We did behavior in 2 parts. So we did an instantaneous scan at each site, and we did a focal survey. So for our instantaneous scan, whenever we arrived at the site, we would scan the flock and record the behaviors of each individual, what it was doing in that instant. And then for the focal surveys, we watched one bird for 5 minutes or until it flew away, which happened quite often. But for as long as we could, we'd watch it and say everything that one bird was doing. And we were really interested in foraging behavior in particular, because again, this is all tying back to the invertebrate availability that we have in the lake. So the 5 foraging behaviors that we broke down were surface picking. So this is where the bird is just getting something either off or right at the surface, and we just see the bill enter the water. Spinning, which is the characteristic phalarope spinning like a top on the water to upwell prey from below it. Air picking, getting something out of the air. Dunking, which is what most of the ones in this image are doing. They've got their whole head submerged. And then vertical bobbing, where they tip up like a dabbling duck would. We also recorded non-foraging behaviors, and so this included movement, such as swimming and walking, alert, simply looking around, preening, or inactive. And then we took from those movement behaviors, so in any given flock, when we were doing an instantaneous scan, there would be birds that were moving that we knew from a focal survey that they were also foraging. So you saw it swimming, but that was in between surface picks. And so we calculated the percent that were not swimming, that were foraging, and multiplied that by the number that were swimming to kind of get that number that were swimming that were probably also foraging. And we considered those searching, so that's kind of our 6th foraging category is birds that were searching for food. So this is broken down by our different sites and our different years. And so you can see, for instance, in Carrington, we had kind of an increase in proportion of the flock, and this is for the instantaneous scans. So all of these percentages are proportion of the flock that was engaged in that behavior. So we saw an increase in birds that were engaged in foraging behavior and the deep Carrington sites in 2015. Pretty similar for the 2 years in Gilbert, low proportion of the flock relative to some of the other sites that were engaged in foraging behavior. And then in Farmington, quite high at the deep sites and at the shallow site. Ogden, certainly more were engaged in the foraging behavior in the deep sites than the shallow. You'll note that these for Ogden are 2014, and the map I showed you earlier only had 2013 sites for phalaropes. We did have one location, and I think you can see it. I'll go back to it, yeah. This site here, this site here is where these observations were done from in 2014. So those are in Ogden Bay, not in Farmington Bay. And that was right at the beginning of the summer. That was in June 2014, where there was still enough water, apparently, for them to use that area. So that's where both of those figures come from, was one day at that site. And so I reordered these so that you can kind of see which sites were most important and when. So Farmington Bay is a really important area for these birds, and I think this is an important result to highlight because Farmington Bay is not super well known. We have done more research in the wetlands and kind of, the needs of ducks in those areas, but as for the main part of Farmington Bay, we just don't know a lot about it, but clearly it's a really important area to phalaropes, both for the shallow areas and the deep areas. And then again, Ogden, in the deep areas of Ogden at least, when phalaropes were there, again, a really high proportion of the flock was foraging there. There's a lot of numbers up here, but I want to highlight a couple things. This breaks down all of the foraging behaviors into the different types. So what proportion of the flock that was foraging was engaged in each of these foraging behaviors. So you can see, for instance, in Carrington Bay, we have a really high proportion that were surface picking. Now, this makes sense because in Carrington Bay is where we have a lot of brine fly adults, so they're typically sitting on the surface of the water. So those are the ones that we're seeing the birds that are there be picking at the surface. Gilbert Bay, a little bit more of a mix, some surface picking, a little bit in here, but then we see a lot searching. Gilbert Bay is typically where we see lower densities of invertebrates, and so birds are spending more time doing this swimming around, maybe searching for prey, and less time actually eating prey. In Farmington, we see much more diversity. We see a lot more spinning. Again, in these shallow areas, they're very commonly dunking behavior. And then in Ogden Bay is where we see the greatest diversity. We had some all across the different categories. But in particular, something I wanted to highlight was this difference that we see in all of the sites from 2014 to 2015. If we look at this category for the proportion of the flock that was searching rather than actively feeding at any given time, we had an increase at every site of more than double the proportion of the flock that was searching. So this is where it'll be really interesting once I do get the invertebrate data to be analyzed. It's kind of like a to be continued, stay tuned. It'll be interesting to see whether there was a decrease overall in prey, whether there was a decrease in a particular type of prey that phalaropes were looking for, because again, there was just that increase in searching time as opposed to actually active feeding behaviors. And then I went ahead and ranked each of the sites based on, so a high searching rank means that there was a smaller proportion of the flock searching, a higher proportion of the flock that was actually engaged in active feeding behaviors. And then foraging is the proportion that was in foraging behaviors. So you can see for Farmington shallow areas, it's pretty similar both ways, but for Carrington deep, even though a low proportion of the flock was engaged in foraging behaviors, those that were mostly feeding rather than foraging. So again, this kind of points out to some important areas. Again though, note that that was 2014. So we have one site in 2015 that ranked really high based on this ranking system, but then we had 3 sites in 2014. So again, this just kind of indicates some changes that may be.
I'm sorry, would you restate that on the search rank being number 2 means what? There's less birds?
Fewer birds that are searching and more that are actively feeding.
All right,
yeah.
thank
So in
you.
other words, a higher proportion that are probably being successful in finding something there. Yeah. All right. Any other questions this far before I move on to the next part? Okay. As for what phalaropes are eating, so our methods for this were simply to conduct lethal collections from various areas of the lake. And we did this in 2012 a little bit, just to figure out what the heck we were doing, and then 2013 through 2015. The original plan was to do Carrington and Ogden and Farmington all 3 years, and then we lost Ogden. So in 2014, we also sampled some birds from Gilbert, and by 2015, we had a pretty good idea of what birds were eating in the different areas. So instead, we chose to focus only on collections from Farmington, but get a better idea of how prey might be changing throughout the season. So these were done, there were collections done early and late in the season, again, early and late, and this is for both species. But then in 2015, we really wanted to look a little finer scale as to how that might be changing throughout the time period that the birds were there. Again, I think these results are telling of something that we may have lost with Ogden Bay no longer being used by phalaropes. So in 2012, all the birds we collected from Ogden were red-necked phalaropes, and 100% of the birds had food in their upper digestive tracts. So for all of these results, we looked at food that was in the bird's upper digestive tract, but also what was in the gizzard, just because we were trying to figure out what they were eating in general. But the upper digestive tracts are especially useful because once the food hits the gizzard, you may be biased towards towards harder to digest items, so you can't really get an idea of the proportion of things they're eating. So having food in the upper digestive tract is really useful to us. I also think that having a higher proportion of birds with the food in the upper digestive tract, that indicates those birds had just been eating too. This isn't they ate this a while ago, this is they just ate this. And so Ogden, we had 100% of birds that had that prey in their upper digestive tract. 100% of birds had food in them, and all of them had been eating, 100% had food in them, 87.5% had it in their upper digestive tract, and all of those that had prey had been eating brine fly adults. So again, we've done quite a few studies on brine shrimp at this point, but brine flies are much less studied, and we have quite a few gaps in our knowledge of them. But clearly this is a really important resource for phalaropes.
Maureen, what's your sample size of birds that you...
For this one in 2012, since this was our first year, I think we had maybe 30, and then each of the years after that, we had 2013 we had 100 of each species, and then 2014 and 2015 each we had 200 of each species. So for 2013, these percentages are shown as a proportion of birds that contain food. So, and they could have contained multiple prey items, so that's why they don't add to 100. So of the birds, of the Wilson's phalaropes collected from Carrington Bay that contained brine fly adults, 100% of them had brine fly adults in them. Of the Wilson's phalaropes in Carrington Bay that contained food, 22% had cysts and 6% had seeds. So just looking at Carrington Bay, we have, again, the importance of brine fly adults for both species. So 100% of Wilson's phalaropes, 100% of red-necked phalaropes. Some other life stages of brine flies were also used, so we have larvae and pupae that were also used by both species of birds. And then we kind of see similar values of cysts. And the cysts, we're not quite as sure about. This may simply be that they're stuck on the brine flies. Maybe if there's enough brine flies that have cysts stuck on their toes, that maybe that's why they're getting them. They don't necessarily have high nutritional values, so we're not entirely sure why the phalaropes have kind of medium-high percentages of cysts in them. But for Carrington Bay, so again, we see kind of similar between the two birds, and one of the main things that we were looking for, so earlier I mentioned the brine shrimp, and that we just didn't know whether that was a resource being used by the phalaropes, and we can see from this that it's not. We didn't find any phalaropes that had been eating brine shrimp. In Ogden Bay, we had a really high proportion of brine fly larvae that were found inside phalaropes. And again, just really to emphasize the importance of Ogden Bay, 76% of the ones collected from Ogden Bay had food in their upper digestive tract, again indicating that they had just eaten, which was much higher than we saw in these other sites. So again, Ogden Bay was a really important resource from that standpoint. It's also important because there were some captive studies done with red-necked phalaropes where they were offered different stages of brine fly larvae. So they were offered either adults or pupae or larvae, and they all preferred larvae. All the birds that were tested all preferred larvae. So when we see here in Carrington Bay that there's a higher proportion that were eating adults, that may just be because there were tons and tons of brine fly adults and that's more available to them. But apparently when they're available and when they can get those brine fly larvae, they really like that. And in Ogden Bay, that seems to be where they were able to do so. Finally, for Farmington Bay, this was a little bit small in terms of birds that had food. Farmington Bay in 2013 was our lowest proportion of birds that had food in their upper digestive tract. And those that did had brine fly larvae, which are not occurring in Farmington Bay super often. So but it looks like when they're there and when they can find them, they definitely try to eat them.
So would the seeds have come from airborne distribution onto the water, or?
They may have, I think they may also just have been, that they got them, because this is including things that were in their gizzards and in their upper digestive tract, so I think it may have just been a long retention time, that maybe that morning that bird had eaten a seed in Farmington Bay, and then it had flown to Carrington Bay where we collected it there. So I think that's probably what we're seeing there. But they also could have floated over there, that's very probable too. 2014, this is what we have from the birds that we've dissected so far. Again, 400 birds per year is a lot to get through, so those are still in progress. But the ones that we have gone through so far from Farmington. So I really want to use this to highlight the importance of Farmington, and again, just kind of a reminder of how little we know about it, and this illustrates its importance to both of these species. brine fly, different stages of brine flies, when they're available, redneck phalaropes especially, we'll use those in Farmington Bay. But Wilson's phalaropes in Farmington Bay really focus on chironomid larvae, so the midge larvae that we find in Farmington Bay that we really don't know much about. As I looked through the literature, I don't even know if I can pinpoint what species they are, or whether there's multiple species. We certainly find them in different colors, and so they may be different. They look a little bit different. So there may be different species out there, but that's a really important resource for Wilson's phalaropes. Also quite a few eating seeds. So again, when those seeds are there, which is a lot more common in Farmington Bay, the birds are eating them, and they're typically bulrush seeds. So both species were eating those. Of the species that we find, of the invertebrate species that we find in Farmington Bay, redneck phalaropes really focused on carixids. So those can be kind of seasonally, we'll get these really dense populations of carixids, water boatmen in Farmington Bay, and redneck phalaropes seem to take advantage of those.
Does that suggest to you that they're in more freshwater systems eating, or?
Yeah, so the carixids will definitely hang out right in Farmington Bay. Like all of these can be, and these are samples from Farmington Bay where we found all those things. So I think they'll be in some of the fresher areas of Farmington Bay, but we found chironomids even kind of farther north in Farmington Bay. So I think they could just take advantage of them there too. So. All right, phalarope physiology, I wanted to just go over our methods for this really quick to give you an idea of where it would go, but I don't have data for this yet. So again, we're using those birds that we had already collected for my third objective. So total, that gives us about 500 birds of each species, which gives us a really great sample size to look at any differences between the physiology. And what we're doing with those birds is measuring their digestive organs. So we want to see, again, differences between the two species and look at changes that may occur over the time period that they're here. Just to see whether there are any differences in between that. And then finally, this is the fifth objective is regarding eared grebe migration, and this is my most complete of my 5 chapters for my dissertation. Eared grebes, we know quite a bit about what they're doing while they're here. We know what they're eating and approximately what areas they're using, but what we don't know is why they're leaving when they do in the fall, whether there's anything that's kind of triggering their fall migration. And so to look at this, we used weather radar to visualize their migration. They show up quite nicely on weather radar from the NexRad station that's located on Promontory Point, and that data has been collected since 1995. So we had data from 1999 through 2014 to look at this migration. And we also compared this to environmental data. So this part I do have invertebrate data for, thanks to GSLEP, who has been collecting data on stages of brine shrimp throughout the lake for years. So that's what we have, that's our starting point. Our 1999 forward starting point is when we have the brine shrimp density data from. So we're comparing the prairie density, the weather, and the lake temperature. And then the 3 things that we really wanted to evaluate with this were first simply the timing of migration. So we can see on the radar when these birds are leaving, and we wanted to look at that across the different years. We've got 16 years of data and wanted to see whether there's any trends, whether just kind of what the average day of migration is. And then we wanted to look at the variables that were associated with departure likelihood on any given night. So on a given night, what makes a grebe decide that they're going to migrate or not? And finally, we wanted to look at some thresholds related to departure. So this, in particular, we're really looking at a wind threshold and a shrimp threshold. The wind threshold is because grebes are not particularly good flyers. They've got kind of short, stocky bodies and short wings, and so they're not great at flying, and so migration can be kind of a dangerous time for them. And so we wanted to see, in particular, whether there was a wind speed above which grebes would not depart from Great Salt Lake. The other threshold that we wanted to look at was brine shrimp densities, because previous research has indicated that grebes will leave from Great Salt Lake when brine shrimp hit a certain density, and that kind of triggers them to leave. So we wanted to look at that also. I wanted to show this image to show what it looks like when grebes are departing on the radar. So KMTX is the radar station that's located on Promontory Point, and then kind of to orient, you've got I-15 and I-80, and there go the grebes, and they look like that, and I think it's pretty fascinating that they show up that nicely on the radar. And so one thing to notice too, I think, when you see this is the size of this flock. So if you compare eared grebes to the rest of the lake, that's 30 miles long of eared grebes that are departing from Great Salt Lake. So when you've got 1 to 4.5 million that are here staging, that's a lot that have to leave. So I think it's a neat image to show really just the magnitude of the birds that are here and their migration.
That's Skull Valley?
That they're going through here, yeah, because this is the ochres and the Stansburys, if I got that right, yeah.
So is it typical they would all leave at once, or does this occur over a couple of days?
This occurs over a couple days, yeah, so that's actually,
So there might be
thank you.
2 or 3. Oh, okay.
Yeah, so groups typically start leaving over this time period that we looked at. They typically start on December 2nd and are finished by January 3rd, but over that span, there's usually 17 nights on average that we see them depart. And we had, I think, as few as 8 nights over which we saw grebe departures, and as many as, I think, 51 one year that we had this really long span and grebes departing on a lot of different nights. So they're not just all going at once. You can see on the radar when the ducks are leaving, you can see when the swans leave, and those occurred certainly over a shorter time period, but the grebes go for a long time, over several days. And so then we were looking at how this compared the earliest and the latest that we saw. So migration started as early as November 18th one year, possibly even earlier than that. When you get to dates much earlier than that, it's overlapping with the ducks that are leaving, and so it can be hard to pick out grebes versus ducks. And then we saw them start leaving as late as December 19th, so that's a pretty wide swing as to when they're starting these migrations. And then they were finished on average by January 3rd, which we can confirm with aerial surveys. We typically don't see grebes on the lake when we fly aerial surveys in January. And so they're typically done, the earliest we ever saw them finished was December 18th, and the latest though, January 19th. In particular, some of these late migrations can be tricky for them, and we don't have, this has been of interest to look at with downing events, and unfortunately unfortunately and fortunately, from a data analysis standpoint, we really only have 2 downing events that would be captured on radar. So there was one in 1997, or there was a series in 1997, and then the really big one in 2011. It's a good thing too, because that means grebes didn't go down a whole lot, but what I can say from those 1997 ones, we had, I think, a span of 5 or 6 days where there were multiple small downings, and they were all in late January. They were all right at the end of when they were migrating. So this may be birds that have stayed on the lake for a while, but then 2002 there was no downing, so they left really late in the year, but there wasn't a downing. So that's not something that we have a very definitive answer on right now. This is to look at the trend across the years. So the y-axis is the day of the median day of migration. So kind of that midpoint of when they were leaving, and the higher numbers indicate later in the year. So as you look across the years, there's not much of a trend. We were curious to see whether, for instance, we would see a more positive trend due to climate change, whether they're leaving later overall because we have warmer winters. When we do plot a trend line on here, it has a very slight positive, but it's not significant. So there's really a lot of variation across these years as to when grebes are leaving from the lake. We modeled departure likelihood by taking all these different weather variables and prey density variables and just using a generalized linear model to look at, on any given night, what is the likelihood that grebes will depart from Great Salt Lake. And our top model incorporated 3 variables, and that was barometric pressure, a positive association between barometric pressure and departure